Yeah, welcome to yet another contribution to the world of delivery. You've seen a lot of wonderful particles, and I was happy that the one I'm going to talk about today has been largely omitted. So, but there's something which links all these talks together. Whether you are a chemist and make modifications, or designing oligonucleotides for therapeutics and delivery, at one point you have to cross a membrane to have any therapeutic effect. And I don't have to convince anyone in the audience that membranes are really key to biology and compartmentalization and where things end up. And we have set out to work with these membranes on the smallest of scales. And before I move into science, you know where Denmark is, but you probably never heard about Odense. We are sort of a medium-sized university, very much in the middle of Denmark. Lovely place. And H.C. Andersen is probably the most well-known person from our town. But on top on fairy tales, we have very good science. And university campus is about to double in scale in a year from now, when sort of a major hospital, one of the 5 major hospitals in Denmark, is added physically to the university. A very smart move. And with a couple of billion kroner, the university has made sort of a translational building to patch all of that physically together. And we look very much forward to use the fact that everything is under the same roof, from basic science to actual patient. So we look very much forward to that. But you look hopefully forward to biomembranes, and they are literally everywhere, and they have to be crossed all the time, which, as you have seen from the earlier lectures, is way more difficult than we like it to be. And what can you do to help things crossing membranes? There are those which hope in the powers of genetic delivery, which certainly has limitations for some types of oligonucleotides, and for larger ones, I doubt that this will ever be a route for efficient transfer of oligonucleotides. What can you do? There have been people looking at how biology solve these problems, and there are wonderful protein-based mechanisms with snare proteins to control the actual membrane fusion and fission. I will focus on fusion. And is there any way to control that by sort of non-biological means, and actually with oligonucleotides? And why oligonucleotides of all entities to work with? Because they have this wonderful inherent programmability, which comes from the sequence, and that's what we set out to do. In nature, to the best of my knowledge, there are no lipidated oligonucleotides at all. There are a few examples from some RNAs, but these are very isolated examples and not very well documented. But other than that, that has not really happened. So what does nature do? Snare-mediated fusion is sort of a little bit like a zipper on a jacket, where you bring things in proximity and ultimately you cross the barrier to fusion. This is a very fast process, and it limits actually the speed of your thinking. This is very interesting. That's why you can't do anything quicker than on a few milliseconds, because membrane fusion is in the way for your communication. Some of you might be faster, but then you are from another planet. Let's dive into it. What do we want? What's the ultimate goal? What we wanted to do when we set out that? We wanted to help membrane fusion, to program membrane fusion, and ultimately to move from synthetic particles to more relevant biological particles. And ultimately to load them, to have cargo transfer. And this is the first major difference to when you talk about this with a biophysicist. For a biophysicist, membrane fusion is already when you start mixing lipids from different membranes. Then they already start talking about fusion. And that has caused a lot of confusion among the community when early reports by biophysicists talked about membrane fusion. And of course, people have tried oligonucleotides before and reported on membrane fusion. But what we are interested in as a community on therapeutic oligonucleotides is content mixing, content transfer, and not just mixing lipid membranes without any content transfer. And that's what we set out to do, and develop what we call lipidated oligonucleotides, in short, LINA. And we have, of course, several generations of membrane anchors. The first one looking a little bit complicated, and synthetically they are. And we have moved over to what we call internally third generation, and they are available in relatively few steps. You can make them in large quantities, which would be sort of a couple of 100 grams if needed. And they are very flexible because you decide whether you have the same sort of hydrophobic moiety or different ones or die-on. So synthesis is very flexible. I will not go into it today. And we worked with a lot of different oligonucleotides until we managed to actually not just have lipid mixing, but actually have content transfer. And when we started that out, all the oligos reported, some of them could do lipid mixing, but none of them were able to do content transfer. There were in a did recent group some success with a PNA-based system, with the major drawback that there was more leakage than actual content transfer. So for all practical means, that was not very close to biology. What we wanted was a way of fusing biomembranes with efficient content transfer and very little to no practical leakage. And this is what we sort of got at the end. Being chemists, we went all the way up to 50 degrees, which obviously is not very biological for most systems, but 37 works as well. And this has been sort of early results. The interesting part is that there's essentially no leakage. These are the gray bars beyond sort of background leakage. And thanks to sort of the oligonucleotides on the surface, there's even less leakage than for the sort of control particles, which do not have any oligonucleotides on their surface. So this encouraged us a lot, and we optimized that. And the question was, is there any size effect? Is there anything which impacts whether you can fuse those particles or not? And we went all the way from 50 nanometers to 500 to ultimately 25 micrometers. And as it turns out, except for the diffusion limitation of these larger GUV type entities, which are roughly cell size, that works as well. So there's no link in terms of whether you have a very big particle or a very small particle. But keep in mind, all of these particles are entirely sort of synthetic lipid mixtures. They don't have a protein corona or glycocalyx or any of the many things you expect on the surface of a biological particle. But that was a very good start, and we have been very happy that we managed to fuse them. But then I talked about program fusion, and one fusion step you can have by just having oppositely charged particles. They will certainly fuse, but this is not very sort of useful in therapeutic content. So, and then we tried whether we can do 1, 2, 3, 4 steps. And it actually turned out to be possible, and we did that. And we did that with works on enzymes and a little bit of classical chemistry. And this is very well established, and that works. And then we moved on from sort of our classical bulk experiments to something a little bit more sort of looking at individual particles. So we joined with a team in Copenhagen, Nico Shasakis, and did some turf microscopy and looked at a lot of particles. That means roughly 40,000 at once. Whether from all these particles we use, is there a subpopulation which participates in these fusion events, or is all of them, or the differences in kinetic between? And the short story is no. They all join into fusion, and there is essentially only in case of full complementarity. So for mismatches or non-complementary, you don't see any fusion on the time scale. We followed the process. And the interesting part was actually we sort of thought in our heads that most particles will do one round of fusion and then they run out of interest in doing another one, but it's actually not the case. 1, 2, or even 3 rounds of fusions by sort of statistically 1 linear pair is something you see. Interesting is based on these measurements, because we have so much data on one set of particles, we can do some statistics, and you only need 5 double-stranded linas to make 1 fusion event between 2 particles. So these are actually really, really, really small amounts of material you need to do this. There are also fusion systems based on peptides. They are non-programmable, but they lose orders of magnitude more material, so they are not very interesting. If you need more material for the fusion, then you would have therapeutic cargo. It's not a very good formulation. So here oligos really show what they are capable of doing, and we were very happy. And but now we'll come to the more interesting part of the story, and this is, can you actually do those fusion events on actual biological particles? And we had a very short discussion on extracellular vesicles, and they are wonderful, and they received the well-deserved interest of a broad field of researchers, and they are known for ages, but I think it's first now that we really understand that they are a major part of biology with very, very different functionalities. There are good ones and bad ones, but one thing is for sure, they are like mini cells. They have a very complex structure. They carry genetic material, they carry proteins, receptors, and what is most interesting for the therapeutic community, they are very good at crossing biological barriers. And that includes the most difficult one, like blood-brain barrier. And we have seen there have been wonderful results for oligonucleotides on neurological diseases, but spinal injections is really not a very sort of patient-compliant way of delivering oligonucleotides to the brain. So it would be wonderful if there would be sort of more regular formulations to solve and really to harvest the potential of those oligonucleotides. And that's what we try to do. They come essentially from all tissues and cell types with a very, very few exceptions. And they differ in size, and some of them are called exosomes. So these are the ones which are sort of in a smaller, so they are under 150 to 80. They are slightly different definitions, but these are sort of the smaller subset of extracellular vesicles. And as you can read here, they spread infections and they sort of work on long distance communication. They are very interesting. But one thing is totally lacking. You cannot change what they carry. You cannot easily cargo load them. There are these good old brute force methods from molecular biology, electroporation, co-incubation, and all of this where you have no control whatsoever. And in most cases, electroporation, you will lose the content, you will scramble things from the inside to the out. So what you have at the end is certainly not a biological particle. What was needed was a very mild method to load sort of cargo and to use these particles in the context of delivery. And that's what we tried. So we used synthetic ones, which have a wonderful surface, which is fully naked, except for the linas we have on top. And then you have sort of these exosome type lipid particles, which have all sort of proteins and glycocalyx and whatnot. And especially the content is interesting because this offers some applications in terms of, for example, liquid biopsies. Cancer types, they will all sort of produce these exosomes, and they may contain microRNAs or other markers telling their activities, maybe prepare other tissue regions for metastasis and whatnot. And that will be very interesting, and that would probably be early onset disease markers. So it would be wonderful if we could test them. But often these RNA amounts are so tiny that classical RNA isolation methods, you will not see it in the ocean of other RNA, and they will long be degraded until you can measure them. If you would be able to get into an exosome and have some sort of readout without unpacking them, that would be a lot more convenient. I will limit myself to the sort of therapeutic attempt, and that's what we did together with a group in Aarhus, Jörn Krebs. He isolated a particular subpopulation, and we sort of designed the oligonucleotides and prepared the synthetic liposomes with sort of the cargo, and did all the bulk experiments to be sure that fusion works. And the outcome is fusion did work, so much I can disclose. We did that sort of surface immobilized that we could sort of wash off anything which hasn't fused with the exosomes. The oligonucleotides down here are also lipidated oligonucleotides, so they're sequence-specific captured, and in a two-hole release, you can sort of the cargo-loaded exosomes can be released, collected, and further up concentrated, and of course characterized. Very useful. And what is the ultimate outcome of that? There are a lot of controls. This is, of course, the lovely lipofectamine, which is a very good standard. And this will always outperform most other techniques because it just goes in. But we all know this is very useful in cell studies, but it's a dead end for any drug delivery. The slide red M-cherry signal over here is the outcome of us loading liposomes with M-cherry. Why M-cherry? It's roughly 300 kilodalton in size. So it's, if you can deliver M-cherry, you can deliver any oligonucleotide, any protein of a similar size will always be likely be encapsulated and delivered with a similar to a similar extent. So this is actually the first example and has been published last year. And we have continued to work on those because you might be lucky with one subpopulation of these exosomes. We have spent our time on browsing as many exosomes as we could get our fingers on. And it's not published yet, but at least for most of them, we can do this fusion. So it seems, as far as we know so far, a generic process that we can fuse those synthetic liposomes with exosomes and load them with various cargos. So we have done that for oligonucleotides, for enzymes and proteins, and work with those. Interesting in many formulations would actually be to deliver more than one thing. There's the wonderful chemistry community, and I'm a chemist myself, so I'm allowed to say that. Conjugates are wonderful, but they are very cumbersome. And as soon as you try to combine more than two things together, this is typically very low yielding. It's not very easy to test different combinations. And the worst thing of all is chemistry is stoichiometric. So if you combine A with B, you will have a ratio of 1-to-1, very unbiological. In many cases, you will need a receptor ligand, let's say 1, and you need 100 times more target cargo for your therapeutic effect. How do you do that chemically to combine things 1-to-100 ratio? That's, you can do dendrimers and all sorts of things, but it's certainly not practical. And as soon as a third thing comes in, this gets completely impossible to study. So it would be nice to be able to do multiple rounds of fusions where you combine things in your target particles at a ratio at your will, without actually chemically combining them. And many of these functionalities, there's no reason that they are linked. Even the wonderful GalNAc is for targeting, but once you are at a site, why is the GalNAc still attached to the oligonucleotide? There's no reason for that. So it would be much better, you would deliver things with all the functionalities you need, but not necessarily covalently linked to each other. So we have just, I'm only showing one example. We have done that for a number of exosomes, and the graphs look very similar. There's a different kinetic and a different efficiency, but those who actually are amenable to fusion will look like that. So in this case, we have used SRB. This is only for SE reasons. It's a self-quenching dye, which lights up. This will be the last scientific slide. And lights up upon dilution. So if you fuse that, it lights up because the self-quenching dye gets less. And for the next step, it's the same. That's why the signal goes a little bit down, but it's as efficient as in the first step, but it's a different dilution scheme. You see, if you follow that on NTA, nanoparticle tracking systems, and you follow the size and so on, you can see that the EVLs grow in comparison to the particles you start from, which makes sense if you do fusion. So that's it for what we have done so far. So you can fuse natural particles. We extend that to cells and all other particles and see how far we can go. What I haven't talked about, and which is super important for the chemistry community, you need a lot of design for those oligonucleotides. So it's not just adding a hydrophobic unit to an oligonucleotide and then you go. There are a few people which I wish to thank. This is Nazmir in my own group. She is a PhD which has almost finished and has done most of the work on exosomes. Metamela, which I collaborate for many, many years, and she's actually here 2 times because we started collaborating when she was a PhD in Niko's group. Now she has become a whole new person in Jörn Krem's group, and she is certainly moving on. And they have contributed most to this. A few founding agents, and of course you have to prepare for such a lecture, and I'm happy to take questions in case there is still time for it.